Okay, cell biology fans, we're going to talk today about cell junctions. Everything we've talked about so far has really been about what's inside the cell, and we're beginning to get to the place where we can start talking about how cells communicate with one another. That's through cell-cell interactions, and how cells are linked to materials outside of the cell, and that's the extracellular matrix, or ECM. So this lecture will be about the cell junctions. These are all proteins that are proteins that the cell makes and allows for the interaction between cells with one another and with the material outside of cells. If you think about how the human body is made up, we are made up of tissues and organs, and those are all made up of cells. All of life is made up of cells. But there's other stuff that allows for those cells to come together. And it's that other stuff, the plus stuff, that we're beginning to start talking about. Nerve cells interact with one another. Muscle cells are bundled together. Even the blood is considered a tissue because blood and the blood cells that are involved in your body are interacting with one another. You have lymphoid tissue in your lymph nodes, in your spleen, in your thymus, and all of your epithelial cells and connective tissues, those are all linked to one another. So if you think about this in a way that gives you the concept of, okay, cells are cells, they're little individual living beings, but those cells then have to interact with one another, right? People are individuals, but we all come together as a community, as a living life force to be what's going, right? So there's all different kinds of living beings on the earth, and we interact with one another in certain ways. I'm going to start off with these pictures because the pictures tell you a lot about what's going on. Let's start on the bottom right-hand side here. If you look at this image, you can see a bunch of cells, right? So you can see, I'll just point to some cells. And the cells seem to be coated with something. Even on the bottom here, you can see a bunch of cells. And they're interacting with one another on this BL is the basal lamina. So cells are going to interact with one another, but they're also going to interact with molecules that have been excreted or produced through the process of exocytosis, and they interact in that way. In the top left image, I love this image. This is a fibroblast cell that I'm pointing to, and it is sitting pretty much by itself in the tissue that it's in, and it's surrounded by extracellular matrix molecules that we will talk about in the extracellular matrix portion. And these are mostly collagen fibrils. So cells have to interact with one another, but they also have to interact with molecules that are extracellular. Here's another image to get the point across that cells aren't by themselves. On the left, you see some cells. If I draw, right, here's a cell. Don't really know where it starts and ends sometimes, right? So I can't really tell if that process is part of that cell. Okay, but you get the idea that these cells might have little places where they interact with one another, right? And then they also have a lot of interactions that might be with materials outside of the cell. In the panel on the right, okay, this is a scanning electron micrograph image, right? It's cutting through cells. You see a lot of white space. That white space is not really space, all right? It's extracellular matrix molecules, and that's having a lot of interactions with the cells. And on the right, if we go back to the image on the left, you see that is a I'm sorry, I have that backwards. The image on the right is a TEM, sorry, transmission, and this run on the left is the scanning electron micrograph. Right, so the scanning shows you the 3D structure, the outside of cells, whereas the TEM, the transmission, does the slice in through the cells. In both images, you see that the cells, maybe they look like they're alone on the right, but they're not. They're interacting with that extracellular matrix that doesn't show up on EM. And you should know now by what doesn't show up on EM, right? What's electron dense and what's not electron dense. And then you can really see that there's tons and tons of interactions that help these cells become the tissues and organs that we know function as far as making you be alive. Okay, let's go back for just 1 second and talk about epithelial tissues, right? We talked about the fact that we have polarized cells, and if I draw for you my typical polarized cell, I make a little square with the squiggles. The squiggles are cilia or microvilli, and these cells sit in a particular way that allows them to interact with one another in very regulated fashions. We know that the apical side, right, the apical side is here, and the basolateral side, basal and laterals, right? So these are the basolateral sides. Maybe I should have actually drawn these red and blue. And that they're actually separated, the two sides are separated, and that separation in sides is really important for how these cells function, because we sometimes want molecules to come in the apical side and out the basolateral side, whereas other times we might want something to come in the basolateral side and go out the apical side. All right? And in general, there's not a lot of motion that's going between the cells. I'll use black for between the cells. So between the cells versus through the cells. Remember, through the cells is called transcytosis. That's a cross, trans. And the between the cells here, that is called paracellular motion. Paracellular is between. Another word for transcytosis is transcellular. Oh, excuse me. And so you'll see these words, and you need to know what each one of them means. Okay, so in epithelial cells we have different sides, and we're going to have them interacting with one another. And that leads us to this image where you can see, right, the issue of different spaces between the extracellular or the intracellular sides of a cell. So in this case, there are two different extracellular spaces. There is the basolateral, right, the basolateral extracellular space. Here is shown with little green molecules floating around in it. And then there is the apical extracellular space, right, apical extracellular. And they're showing you little red molecules that are up in the extracellular space of the apical side, and that we can have transcytosis occurring through the different early endosomes that are part of the apical early endosomes. And the late endosome is where we think that both the basolateral and the apical sides meet, and there's some sort of processing there that allows for transport to its final destination, whether it should be apical, basolateral, or the lysosome. So you can see both green and red in the lysosome. But some of these molecules might actually be exocytosed, right? So something that comes in from the apical side might actually have to get out on the basolateral side, and vice versa. Something taken in on the basolateral side might have to get out on the extracellular side of the apical region. So it's very important that we have these two different sides, and the junction that we're going to talk about with respect to what delineates or what describes what makes the two sides different is this tight junction. One more example to show you is if we look at tissues in a particular region, the tissues of the gut are really important for obtaining nutrients. And the way this works is that there are gradients that allow for the movement of nutrients across cells. So let's just take a look at this picture. Here on the top part of this image is the lumen of the gut. That is either your stomach or in your intestines. It's the lumen of the small intestine where most of our nutrient absorption occurs. And there are epithelial cells that line the lumen, and it's actually the apical side is here. If I were to draw, right, these are all apical, so these all have microvilli and cilia to increase surface area for the uptake of nutrients. And so nutrients get taken into the and through cells through the process of transcytosis, and they end up being taken up into usually blood vessels, which they're not showing you here. So there's a lot of blood vessels that are, right, these are not big veins or arteries, these are little tiny venules and little tiny capillaries, but they allow for the uptake of these nutrients into your bloodstream. But underneath the epithelial cells, we have something that's called connective tissues. And those connective tissues, they're connecting the epithelial cells and the underlying cells, which are smooth muscle cells. So they're showing you have smooth muscle cells that go in two different directions. These cells are going sideways like this, and the cells that are underneath here are going long ways, okay? And what you're seeing is the very end of cells. So you have these smooth muscle circular fibers and longitudinal fibers, and they allow for the expansion and contraction of these tissues. And then you have a little more connective tissue. And it's interesting because the connective tissue has very few cells, but it has a lot of extracellular matrix molecules. So I just circled, right, these little things with the dots are the cells, and those dots are supposedly the nuclei, but all of the material outside of that is extracellular matrix. Same thing down here in this connective tissue. And then there's another layer of epithelial cells at the very bottom. So your tissues are really composed of a lot of different kinds of cells. And these different kinds of cells allow for the overall function of any particular tissue or organ in the body. Here's another example closer up of the gut and what happens. We have these epithelial cells that line, right, the lumen of the gut, which is shown in orange. So this is the inside of your intestine. And glucose is one of the main things we need to take up. There are glucose transporters on these epithelial cells that allow glucose to be taken into the cell. It is taken directly into the cytosol. And what happens is it's at a high concentration in the cytosol, and so we know that molecules can go from a high to a low concentration, and there are different transporters that allow the glucose out into the blood vessels that I already mentioned that are in the extracellular space underneath these epithelial cells. In the very top portion here, right here's the tight junction, and what the tight junction does is prohibit that paracellular transport between cells, which we don't really want because you don't want stuff just leaking in. And glucose in particular, it's interesting, so glucose is low in concentration in the extracellular apical side, and right, we know that it moves in this direction, so it goes from low to high. So that's these green molecules are pumps that go against the concentration gradient, whereas these red molecules on the basolateral region are not pumps, they are transporters or channels because they go with the concentration gradient. Okay, so just be sure that you understand the issue, right? Glucose is taken through the cell, which is transcellular or transcytosis, whereas molecules that go between cells is paracellular transport. So the cells that we are going to talk about all cells form selective permeability barriers. So that means that only certain molecules, right, selective, and we say certain molecules, the ones that are allowed, right? So who goes there? Halt, who goes there? And if you have the right credentials, you can pass. So this means they must be wanted or needed in the cell. Wanted or needed. Okay, but cells form a permeability barrier, and the movement of molecules through or into cells is regulated, some of it by cell junctions, some of it by transporters. And what we're going to talk about here are the cell junctions that allow some of the features that we're talking about in larger organizations of cells, tissues, and organs. So I usually showed this as a framework for you to think about, okay? There's 3 kinds of cell junctions. The first one that we'll talk about are occluding junctions. So if you think of occluding, okay, occlude means to stop, right? So think of a stop sign. How many sides does a stop sign have on it, right? I think it's 8. I won't be able to draw that very well, okay? But you get the idea. An occluding junction is stopping something from going from one place to another. And we're going to talk about 2 of those, tight junctions and vertebrates, and I'm only going to very briefly mention what septate junctions. Those are invertebrates. So these are the molecules that prohibit things from going from one place to another. Then I'm going to talk to you about anchoring junctions. So okay, you know my drawings, not very good, but I think of an anchor looking something like this, right? And so what does an anchor do? It holds things down. And anchors are going to allow us to hold cells to cells and cells to extracellular matrix. There's 2, and they're going to 2 different sets, so there's 4 total here. All right. In the occluding junctions, we had only 2. All right. And then in the last part, we're going to talk about communicating junctions, and this is talking one person to the next, one cell to the next. How do cells communicate with one another, right? Usually you can communicate in multiple ways, and that's what happens in different kinds of cell types. So we're going to talk about gap junctions. We've already talked a lot about chemical synapses, and briefly I'm going to mention what happens in plant cells and plasmodesmata. So let's start with the occluding junctions, and I already said occlude. All right, if you have an occluded blood vessel, you're in big trouble. That means that the blood isn't moving through it. Okay, so occluding means to block. And no transport, okay? And I should have used red. I think of red as stop, right? So occlude means stop. And tight junctions, all right, have multiple functions. They keep macromolecules from going between cells, right? If I draw for you my typical epithelial or endothelial cells, the tight junctions are up here at the top, and it precludes, okay, it stops that paracellular transport. Another interesting feature here, though, is that the tight junctions also keep the apical region apical and the basolateral region basolateral, which means that proteins that are in the apical region can't then, in the apical membranes, can't migrate to the basolateral membranes, and proteins that are in the basolateral membranes can't migrate to the apical region. So it's like a fence, all right? It's stopping the movement of molecules from one membrane to another. So those are the 2 functions. This image shows you a really nice EM image, and they've shown you the cartoon in figure A on the left. So you're looking at the microvilli where the red arrows are, and you can see some microvilli here, 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 and then underneath the microvilli are where the tight junctions are located. So in the cartoon, the tight junctions are proteins that are embedded in the membranes that allow for, I think of this as you have 2 cubes that are next to one another, and you're if you don't want anything to pass between them, you have multiple tight junctions. Notice they're showing you, right, tight junction, anywhere these two membranes touch. Maybe if I make that bigger, you'll be able to see it a little better. So, actually, let me do blue. Everywhere these guys touch is a tight junction. So, and there's no set number of how many tight junction interactions there are, but what happens is when you look at that on the EM image, okay, and this is a freeze fracture EM image. How can I tell? Because it talks about the E face and the P face. And in here, all of these lines, maybe that's not the best color, all of the little lines that are up here are tight junctions that are prohibiting movement of molecules between these cells. So you need to understand what the tight junction's functions are, but you also need to understand what it's made up of. And it's made up of proteins. There's 3 proteins that I'd like you to know. It actually has more associated proteins, just like everything we talk about. It's more complicated than we can really go into. So I'd like you to know just the bare bones here. And the way these junctions, almost all the junctions work, is that there are transmembrane proteins, and then there are some peripheral intracellular proteins. And so the transmembrane proteins that are part of tight junctions are called claudins and occludins. In panel B, shown on the bottom left of this slide, they're showing you that claudins are the dark green molecules that are transmembrane proteins. And how many transmembrane passes does a claudin have? Well, count, right? How many times does it go through the membrane? If I were to maybe highlight these, right, I could say there's 1, 2, 3, 4. So these are 4-pass transmembrane proteins, and you know how 4-pass transmembrane proteins are made. They're even telling you that both the N-terminus and the C-terminus are in the cytosol of the cell. So the N-terminus is in the cytosol. Cytosol. Right? How would that protein be made? That's something that you might want to think about. Claudins and occludins both have a similar structure. The N and the C termini are in the cytosol, and they both are 4-pass transmembrane proteins. Claudins interact with claudins, occludins interact with occludins, and they're doing that across cells. So this space here is this space between cells, and the gray is the cytosol of one inside of the cell, and this is a second cell. So this is cytosol here. So these claudins and occludins can interact with one another and generate, as I'm drawing, right, circling, interactions at any particular place along the cell. And you can see, right, they're showing you that you can have all sorts of interactions, and it can be complicated how these cells interact with one another. Why do you think you might want to have so much tight junctions? Well, think about it this way. If you were to take two sheets, and you sewed them together, all right, how would I do this, right? So here are 2 sheets, and you sew them together with one stitching across the top. Well, if you don't want something to go between those 2 sheets, that one stitching is good right up until the point where one of those places gets weak. So if you have multiple stitches, right, so you have a whole bunch of stitching across the top of these, the chances of anything going through is very small. So there's lots of these proteins. The claudins and occludins are the transmembrane proteins, and they're going to interact on the intracellular side. Okay, let me just take a big blue thing, okay? They're going to interact with these ZO proteins. Okay, on both sides you're going to have ZO proteins that interact with the claudins and occludins, and that's going to be important because the ZO, the peripheral intracellular proteins, are then going to interact with a cytoskeletal component. The cytoskeletal component, so I'd like you to look at panel A, and I'm going to draw, right, so if we just look here at the top portion, what you see are tight junctions, and that's shown in panel B. You have claudins and occludins on both sides, and they're only going to show you then the additional interactions on one side. So on the right-hand side, they're showing you have ZO proteins that bind to the tails of the claudins and occludins. Remember, the N and the C termini are in the cytosol. So the ZO proteins are peripheral transmembrane proteins. Right? Do you remember what peripheral transmembranes, peripheral transmembrane proteins? They interact with transmembrane proteins, and those link to actin filaments. Okay, so actin is going to be interacting with the tight junctions, and if I blow this image up on the right, I know it gets blurry, but what you can see is that all of the red lines up here are actin. So the microvilli have actin. There's actin that's interacting with the tight junctions. And all of that helps to cross-link and create a very stable structure for these epithelial cells. We're also going to see when we talk about the next junction down, they also link to actin. And that actin is also going to link to the actins that are part of the microvilli actin and the actin that's found in tight junctions. So let me zoom back out. Okay, so in this case, right, we're talking about claudins and occludins, claudins and occludins, and ZO proteins, and all of this is going to end up getting linked to actin. Okay? All right, so here's just the words for those ZO proteins, intracellular peripheral membrane proteins that interact with actin. So the claudins and occludins don't interact directly with the actin, but the intracellular peripheral membrane proteins are the link between the two. Now, we're talking about occluding junctions, and I said I would mention occluding junctions that are in invertebrates. So invertebrates, right, vertebrates have tight junctions, whereas invertebrates have these septate junctions. They're similar because they actually occlude the movement of molecules between cells. But if you look at this, this reminds me of a railroad track, and the linkages between the two cells are very regular. And so, not really sure. Sometimes septate junctions come up in standardized exams, but I wanted to mention it to you just so that you had heard the words. Hopefully, if you see junctions and septate, it might ring a bell if it ever comes up in some sort of standardized exams. Of course, if you're going to go and study invertebrates, you're going to learn a lot more about these septate junctions and their importance. Okay, so that's the end of occluding junctions. All right, we had three types of junctions. We had occluding junctions, we have anchoring junctions, and then we have communicating junctions. So type 1 is done, occluding junctions. Now we're going to work on anchoring junctions, and there are 4 different types of anchoring junctions. Let me just see if I can get my red pen. So we're going to talk about 4 different types of junctions. And these junctions are all going to link to an intracellular cytoskeletal filament, 2 of them to actin, and 2 of them to intermediate filaments. And what's nice about these is they're broken down in this, right, in this graph. You can study this graph and try to memorize it, or you can think about how these function and get the information really not memorized, but that you understand it. So in anchoring junctions, we have 2 different types of anchoring junctions. We have cell-cell, which means one cell to another cell, or we have cell, and this says matrix, which means extracellular matrix. Now I know we haven't done the extracellular matrix. As a matter of fact, it's coming up, extracellular. It's coming up just as soon as I'm done with this lecture. But what I'm talking about is cells interacting with molecules that are outside of the cells. Okay, and notice in cell-cell you have one that links to actin and one that links to intermediate filaments, and the same in cell to extracellular matrix, one that links to actin and one that links to intermediate filaments. So let's walk through these, and yes, you're going to have to do a little bit of memorizing, but really try to understand what's going on with these junctions. Just as I said about the occluding junctions, these junctions are going to have some sort of transmembrane protein. Okay, and this one they're showing you the transmembrane protein right here or here. All right, and they're just showing you one transmembrane protein. It's interacting with the blue peripheral transmembrane proteins, and that's linking to some sort of intracellular cytoskeletal filament. So if you get that part down, you're going to be a step ahead of the game. If you know that there's a transmembrane, at least one transmembrane protein, one peripheral membrane protein, and then some cytoskeletal filament, okay? It narrows down what you need to know. We're going to start out talking about the cell-cell, all right? And let's just jump. The first one is called an adherens junction. What do you think of when you hear adherens? Adhering. What is adhere? What does that mean? It means to stick together, right? If two things adhere, they stick together. Very different than occluding, where you're stopping things. Here we're sticking things together. Okay. These are the two cell-cell junctions. Adherens junctions and desmosomes. Desmosomes, you can't get what they are from the name, but they're going to use adherens junctions and desmosomes use proteins that are called cadherins. Okay, I remember this because adherens and cadherins sounds very similar. The cadherins are going to be the transmembrane proteins, and then you're going to have some type of intracellular peripheral membrane protein, and those are going to have another type of name, and then they're going to link to some sort of cytoskeletal protein. The adherens junctions are going to be actin, so A and desmosomes, right, are going to be linked to intermediate filaments. All right, so here we go with the tough ones, because this is going to make it hard for you to sort of remember all the different ones. But the adherens junction has cadherin, is the protein that's transmembrane. So here in this image, the green proteins. So it has a transmembrane region, and then it has multiple globular domains that are linked together. This is all one protein. So this is one protein sequence that's folded up, and it has multiple domains. And notice you have two of them on one cell, and cadherins interact with cadherins on the other cell. The blue proteins are the peripheral transmembrane proteins, okay? And they link from the cadherins to the cytoskeletal filaments, and those proteins are called catenins. So there's cadherins and catenins, and those are all linked to the adherens junctions. And the filament that is being used for adherens junctions is actin. What do you know about actin? Well, actin, we said, was a polar filament. It can be polymerized and depolymerized very quickly, and so it's important for cell shape, and it's also important for movement. So in this case, a lot of these actins are all about maintaining the shape of the cell. In this image right now, we're showing the tight junction at the top, and remember, they're not showing you, but this has links to actin. Okay, I'm going to draw them in for you, since this picture chose not to. All right, and then we also have cadherins. So there's a lot of actin filaments that are at the apical region that separates the basolateral from the apical region of the cell, and this is often called the actin adhesion belt, because you're really creating a huge amount of structure at the very apical region of these cells. So these bundles of actin are in the adhesion belt. Remember, the tight junctions are claudins and occludins and zeoproteins, and they're linked to actin. And the next one down, you get the adherens junctions, and those are what are those? Those are cadherin, sorry, I'm losing it, and catenins, and they're also linked to actin. The adherens junctions are not as tight, not as structurally sound, but they do allow for structural integrity of the cell. They are cell-cell interactions. The occluding junctions are also cell-cell interactions, but they're in that different type. They're in the occluding interactions compared to the anchoring junctions. So remember, anchoring junctions are keeping cells in place. The second of the anchoring junctions, the cell-cell anchoring junction, is called desmosomes, and it's linked to an intermediate filament. And right off the bat, that should put up a flag in your head, because you know intermediate filaments are different than actin. Actin is highly dynamic. It can be made, polymerized and depolymerized very quickly, but intermediate filaments are very structurally sound. They are not polar, meaning they have the same sides on each side of the filament. They're very structurally sound. They're made up of a ton of subunits that are linked together in 8 bundles of protofilaments. And I don't know if you guys know what a rivet is, but these look like little rivets that hold, rivets are what hold metal pieces together. And let me just draw an arrow, okay? So the black portion, the very dark black portion on the right-hand side of this image, is showing you the desmosomes. And the desmosomes are linked to these intermediate filaments, which are emanating out from the desmosomes. Okay, this is the image, okay, and it's blown up. So up above, right, you might even have 2 desmosomes here. Not sure which one. I'm sure this is the one the bottom one is the one that's blown up. But you can have multiple desmosomes linking cells to cells. All right, and here are the proteins that are associated with desmosomes. And yes, you do need to be able to recognize them. Okay, they're going to have transmembrane proteins, and on the right-hand side, they're shown here as these paddles. So these are, right, transmembrane proteins that are going to interact with transmembrane proteins on the other side. So you would also have, let me grab some green here. Okay, sorry. So you're going to have proteins on this side that are interacting. Not a very pretty picture, right? But these guys are going to be in a membrane on the opposite side. So the 4 proteins' names are desmogliin, desmocollin, desmoplakin, and plakoglobin. Oh, that's just painful, I know. All right, but do the best you can. Desmosomes, desmogliin, desmocollin, desmoplakin, desmoglobin. Maybe you want to do it alphabetically. Desmocollin, desmogliin, desmoplakin, plakoglobin. Right, desmosomes. Somehow come up with some way to remember this. Okay, these are the transmembrane proteins. So those are the transmembrane proteins of desmosomes. And on the left-hand side, they're showing you how they interact. So these paddle portions interact with one another in the extracellular space. And on the intracellular side, you have a whole bunch of peripheral membrane proteins, these little pink guys. Okay, and they're not really showing that on the right. And that's what's interacting with, okay, so the little pink guys and these little cross-linking molecules. That then, these are the peripheral membrane proteins. And they are what link the cell-cell proteins, the transmembrane proteins, to intermediate filaments. So you have a ton of proteins. I think what they did was they just said this red stuff is the peripheral transmembrane proteins. And they are saying that those are what link to the intermediate filaments in the inside of cells. Notice on the left-hand side they're saying keratin filaments. Remember, keratin is one of the intermediate filaments that's very important for the structure of cells, very important for the structure of hair, nails, skin, and defects in keratin can have dramatic problems for you. So this is why. These are holding cells together, and if you don't have the ability to hold cells together structurally, it's really not good for your well-being. Okay, so notice I didn't tell you what the names for the peripheral transmembrane proteins were for desmosomes. And I did that for a reason, because you might be on overload at this point, and I don't want your little heads to pop open. So let's just keep going and see if we can push through and get to the end of these junctions, and then we'll come back and tie this all together in a way that I hope you can remember it. So we're still doing anchoring junctions. So these are two more of the anchoring junctions, and they are called focal adhesions and hemidesmosomes. Okay, the hemidesmosomes should make you happy because we just did desmosomes. Hemidesmosomes are going to be almost exactly the same as desmosomes. Okay, but these two anchoring junctions are cell to extracellular matrix. So they're not cell to cell, but they're binding cells to the material that's outside of cells. And this actually is really, really, really important. They're both really important. Linking cell to cell and linking cells to extracellular matrix are some of the things that keep cells from just going all over the place. And the release of some of these junctions is what happens when tumor cells metastasize. Tumor cells normally should have, right, so all the cells in your body should be anchored to the right place. And when they become tumor cells, they become sort of these rogue cells in your body. The anchoring junctions are released, and those cells are then set loose. They're no longer bound cell to cell or cell to extracellular matrix. They can be released, taken up into your bloodstream, lodged someplace else in your body, your brain, your heart, and those secondary tumors are what end up killing you due to the metastases. Okay, so the two other anchoring junctions, one is called a focal adhesion, and that is going to link to actin, and just like the desmosomes linked to intermediate filaments, hemidesmosomes linked to intermediate filaments. Okay, there are some interesting features to these two different kinds of cell to extracellular matrix interactions. In particular, the focal adhesions use proteins that are very interesting called integrins. And the integrins always have transmembrane regions. There's usually two integrins. One is called an alpha and one is a beta. And then there's multiple integrins. There's so many different integrins. Alpha 1, alpha 2, alpha 3, sometimes they have weird names. Beta 1, beta 2, beta this, that. And so there's lots and lots and lots of different focal adhesion complexes that can be made. In this case, all right, these focal adhesions, the integrins, are interacting with extracellular matrix proteins. And we haven't done extracellular matrix yet, but you will see a lot of extracellular matrix proteins like collagen, elastin, and fibronectin are all over the place. In the fluorescence images above, what you see are the intracellular filaments that the integrins are associated with. And in this case, the integrin subunits link through a peripheral transmembrane proteins that have all sorts of different names: alpha-actinin, talin, filamin, vinculin, Okay, you're not going to remember those. You're not remember. Okay, because you can't. I've already given you a lot of things I do want you to remember. So you're going to have the integrins linked to a peripheral transmembrane protein, and we know that because this is a focal adhesion, it's going to link to actin. So in the top left-hand panel, A, you see green, and green is standing for actin. In the top right-hand panel, the red is standing for one of the integrin subunits, either alpha or beta, I don't remember which. And what's really nice is you can see, I hope, okay, if I circle it in blue, you get the overlap of these only at the region where the integrins are actually touching one another. And these are two different cells, and so the cells are being linked together through the focal, I'm sorry, not cell to cell. This is cell to extracellular matrix. So what's happening is you're getting this focal adhesion bound to some substrate, whether it's collagen, elastin, or fibronectin, and cells that are near one another, can, so if you think about, if I look on the right-hand side here, this is probably different cells, one next to another, linking to the, this is saying the basal lamina, whatever molecule outside the cell it's bound to. Okay, so this is trying to, tie this up. Occluding junctions, we did. You should know what those are. Those are, right, stop. This is a stop. These guys anchoring, they're keeping things in place. We had two, right, that are linked to actin filaments, cell-cell. This was called adherens junctions. I can't write very well with this. And the cell to extracellular matrix was the focal adhesion. The two that were intermediate filaments, cell to cell, was a desmosome and hemidesmosome. Did I even talk about hemidesmosomes? No. Because, right, you're already on overload. So I'm trying to minimize what you have to do here. And then the last one that we're going to talk about are the communicating junctions. So the communicating junctions, the one that I'm going to talk about the most are these gap junctions. And if you think about what communication is, right, this is allowing for signals to be shared amongst individuals or individual cells. And communicating junctions are shown here. Large gap junction, small gap junction. On the right, they're showing you an EM, a freeze fracture EM, showing you these dots. The dots are the proteins that are making up the gap junctions, and sometimes you can have really large conglomerations of these proteins bound to one another, or you can have small conglomerations, but they're still going cell to cell. All right, and when I'm sorry, those last two arrows aren't in the right place. Down here, right, where they're actually pointing to. So in this case, what we're talking about is something is allowing these cells to communicate from one cell to the next. I think of these as, if you're on a big cruise boat, there's portholes that you can look out, and those portholes allow, if you were to pass another ship, you could actually pass something from one cell to the next. So this is one ship here on the left, and this is one ship on the right, and these little portholes, those are the proteins that are the gap junction. And the proteins are very cleverly called connexins. I love that. It's so simple. They connect cell to cell. Those connexins are 4-pass transmembrane proteins, and you need 6 of those to make up one individual gap junction. So this allows for small channels, so small molecules, less than 5,000 Daltons. Okay, notice this 5,000 is being blocked. So anything that's smaller than 5,000 Daltons can pass from one cell to another. What's little small things like that? Maybe amino acids, maybe not probably polysaccharides, but monosaccharides, nucleotides. And what's really nice about gap junctions is that in some cells, it really allows the cells to act as one functional unit. And that's really important where you have this process of electrical coupling. Allows for synchronous action, and the obvious example here is the heart. Okay, a heart has to beat. Okay, here's the heart. All right, this is not what a heart looks like at all. But what has to happen is all of these cells have to be synchronized with one another so that it beats all at the same time. Okay, if it doesn't beat at the same time, what would happen? Well, the blood wouldn't go anywhere because you wouldn't have this conglomerated force to push the blood out into the blood vessels. So the synchronicity of this is really important, and gap junctions allow for electrical coupling. All of this is regulated by pH and calcium, and we're not going to talk very much about any of that, but gap junctions are really important for cells that need to function as a unit. That was it. That was all you needed to know for gap junctions. Connexins, 4-pass transmembrane proteins allow for electrical coupling, passage of small molecules, and the communicating junctions in plants are called plasmodesmata. This is the plural. I do believe plasmodesma. Desmata? I'm not sure. Maybe this is the singular and plasmodesma is the plural. These are actual channels between two plant cells, and what that means is, right, here's the cell wall of the plant on the outside here. The cell wall actually stops, and you have these spaces where the cell walls are broken through, and there's a channel. And what's really nice about this channel is it's smooth ER, so they're showing you rough ER on the outside, and then inside here, that smooth ER passes from one cell to the next, so these cells can communicate with one another through the proteins and molecules that are being made, but there's also aqueous channel between these two that allows for the passage of small molecules as well. And if you're a plant person, you'll probably study plasmodesmata in gory detail. Okay, so that's it. That's not a ton. I know it's a lot of names of proteins, and yes, you will have to memorize them and which particular cytoskeletal filament they interact with. But here's a nice summary. You should be able to pretty much draw any or all of these and tell me what the function is. Notice which one's missing. We're missing a focal adhesion, so we should have another one either on the bottom or on the side that links cell to extracellular matrix. And finally, okay, so this picture actually does show you the focal adhesions and the hemidesmosomes. And what I really like about this EM image on the right is at the very top you see the tight junction, the next one down is an adherent junction, the next one down is a desmosome. So we have all of these can occur in one cell, and they really allow for the interaction cell to cell and cell to extracellular matrix. So with that, I will leave you to your own devices. Have a great day.